Hi guys, welcome to my latest video on osmosis. So this is a special form of diffusion and this involves water. I'm going to briefly talk about what osmosis is and show you what happens when water moves through a partially permeable membrane, and then I'm going to direct you to my required practical video related to osmosis in potatoes. Water is important to cells because without it you would not be able to remove waste, bring in nutrients or transport oxygen. Water is found in the vast majority of organisms, and in fact the human body is made up of about 60 to 70% of water. Water can move across cell membranes, and it moves across cell membranes by osmosis. This is the definition that you need to learn here, that osmosis is the diffusion of water from a dilute solution to a concentrated solution through a partially permeable membrane. So the first thing we need to look at is it is a form of diffusion, as it says there, but it's of water. Only water will move through the membrane. And it mentions from a dilute solution to a concentrated solution. So if we look at these pictures of two beakers, the beaker on the right, okay, has blackcurrant cordial and lots of water. So there's lots and lots of water molecules within the solution. Okay, so there are obviously sugar molecules there as well. Okay, so larger sugar molecules. Whereas on the left hand side, if we look at the left side, this is a more concentrated solution. So what we mean by more concentrated is that it still contains water but of a lower concentration, and there's far more sugar molecules in there. So that's what we call a concentrated solution, this one here, and then on the here is the dilute solution. So what we need to understand is how does water move from a dilute solution to a more concentrated solution through the partially permeable membrane. Here we have a cell. Okay, so the outside of the cell, I'm going to say, is the so this is outside the cell, and then you've got the partially permeable membrane, the yellow line, and then into the cell. Okay, so the dilute solution is on the outside of the cell, meaning that there are more water molecules. As you can see, there are more water molecules mixed in with sugar molecules, but there's more water molecules in the dilute solution. In the concentrated solution, there is less water molecules and more sugar molecules in this example. So with the sugar molecules, as you can see, they can move around and they'll hit each other, but they can't get through the partially permeable membrane as they're too large. Whereas the water molecules, they'll collide with each other and eventually will move from where they're in a dilute solution to where they're in a concentrated solution. So more molecules will move from outside to inside, so they'll be moving that way through. Osmosis can have different effects on plant tissue. So this goes back to your work on cells. Animal cells have a cell membrane, but they don't have a cell wall. And a plant cell does have a cell wall, so they react quite differently when they're put in different solutions of water. So a hypotonic solution is when there is lots and lots and lots of water outside the cell. So that is going to be a dilute solution, and there's going to be a concentrated solution within the cell. And what can happen is that water will rush in. Okay, H2O will rush in, and in a animal cell it can actually lead to the cell membrane bursting, and this is called lysis, or known as lysis. Whereas in a plant cell, water will move in, but because of the vacuole, okay, you can't really see the vacuole very well there, it's going to press against the cell wall, and it makes the cells what we call turgid, and that is a normal situation. So plant cells need a lot of water to keep them turgid and firm. In a hypertonic solution, okay, you've got much dilute concentration within the animal cells, and outside very little water. This tends to be, for example, in terms of if the cells are placed into things like salt water, and they will shrivel up because the water is moving out of the cell from a dilute solution to a concentrated solution. With the plant cells, what happens is it can be quite drastic for the plant cells and the plant itself, because the vacuole shrinks, and then it actually pulls away the cell membrane from the cell wall, and it becomes what we call plasmolyzed. And this is when you see plants wilting, for example. They have too little water to keep the cells turgid, and therefore they collapse. The more common thing is when we have what we call an isotonic solution. All isotonic means is there's a balance of water molecules outside and inside. So we've reached kind of an equilibrium. So water will be moving in and out, but at equal rates. So you don't get into this situation or this situation over here. It's worth knowing these things. It doesn't actually state it specifically in the specification, but I think it's worthwhile knowing that for any applied type of questions. Here is part of an exam question, and they sometimes show you this kind of experimental setup. So they say the equipment is below to investigate osmosis. And what they're using here is a piece of what we call visking tubing. Now, if you were to look at visking tubing is a piece of plastic tubing, but it has very small holes in it. You can't see the holes, but the holes are small enough to allow water to move in or out. And you can see that the visking tubing it contains a concentrated salt solution compared to the water outside. So if I just put the water molecules on the outside here, and I've added the salt molecules with the water inside the visking tubing. Now, a common question is to ask you things like, for example, what is osmosis? And then the question will go on to ask you things like what will happen to the water level in the capillary tube during the investigation because of osmosis. Well, with our knowledge of osmosis, we know that water will move from a more dilute concentration to a concentrated solution. So in that case, what will happen is that water will move into the visking tubing. Just show you that. Ooh, not that. So water will move into the visking tubing, and if water moves into the visking tubing, what will happen in the capillary tube? Well, the water level will rise. Okay, so it rises. And the second part of this question, it always will ask you, well, not always, but it mostly will ask you to explain why this happens. So you need to then explain that water will move from a dilute solution, where there is more water outside the visking tubing, inside the visking tubing that contains a solution of salt, which is more concentrated. Osmosis covers one of your required practical activities, and in fact it's required practical activity 3. So hopefully your teachers will have gone through an experiment with you looking at a range of concentrations of salt or sugar solutions on the mass of a plant tissue. In this case, what I used, as you can see in the tubes there, I used potatoes. Okay, and we had 1 molar sugar, 0.5 molar sugar, and distilled water. It's really important that you know this required practical really well, because 15% of your GCSE will cover the required practical activities. Here's a link to my video. I've also put the link in the basic information about this video, so please do go to it and have a look. And it's really good revision to go through the practical activity, and also it gives you some really good tips about analysis of data and how to do the experiment scientifically. I hope you found this useful. My next video is going to be about active transport.